Welcome to this Open Helix Tip of the Week. My name is Jennifer, and I will be comparing microbial resources. For more details about these resources, please see my full post on the Open Helix blog. The first resource I want to talk about is the Ucsc Microbial Genome Browser. Here I'm showing its homepage. For my quick example, I'm going to show you the E. coli K12 display. The display opens, and if you know the Ucsc Genome Browser, you can see that it's based on that. I'm going to use this default area of the E. coli genome to show you the Genome Browser. So we've got our sequence backbone, and along that backbone are aligned all sorts of different information, including the GC percentage, operons, studied loci, all sorts of different features, as well as conservation tracks and relatedness. At the bottom of the browser, you can see that there are track controls. These allow you to turn on and off the information that's on the browser. When I mouse over an item, I get a pop-up information box, and I can use this browser to go deeper and deeper into E. coli information. As a comparison, I next want to take you to the JGI's Integrated Microbial Genomes. The first thing that you notice about this homepage is that there are a lot of tools for finding genomes, genes, and functions rather than lists of genomes. Integrated Microbial Genomes approaches comparative analysis by first allowing the user to select sets of genomes, genes, and functions, and then comparing them and analyzing them. As a comparison, I'm going to do a quick search for E. coli K12 again. I get 2 genomes. I'm going to pick the one that's not a plasmid. Now the page that opens is organism details. It's a huge long page of organismal details. It's not anything like the Ucsc Genome Browser that we saw, which had layer upon layer of information aligned along the genome. To get a similar view as we saw with the Ucsc Genome Browser, I'm going to click on the chromosome viewer, and then I'm going to select a similar region to that which we saw for E. coli using the Ucsc browser. Okay, and what you're going to notice is with this IMG chromosome viewer, it is nothing like the Ucsc Microbial Genome Browser. What we've got is the sequence, and aligned it along it, the only information are these boxes of functions. So I can select what types of functions I want to see, COG, ECG, KEGG, etc. And they are color-coded. Again, I can mouse over for additional information. But as you can see, comparing the Ucsc Genome Browser to the IMG comparative analysis site really is like comparing apples to aardvarks. They both start with the same basic information, namely microbial genomes from RefSeq sequence database, but then approach those genomes and how you analyze and display the information very differently. They're both wonderful resources. Please check them out in more detail, and check out the full text of the post for additional resource comparisons. Thanks for joining me this week.